Good morning or good afternoon. Hey guys, and welcome to another awesome week. We're going to go ahead and start in on topic 6, which is going to be the last topic that we have to talk about. I know this says 7, but that's for IB. So this is another instance where you guys' content overlaps enough that I can use some of their information for you guys. So I'm going to go ahead and use this. But it is Unit 6, and this is the last thing we're going to really talk about before we get to the exam. So without any further ado, let's go ahead and get started. We're going to talk about DNA structure and also its process of replication, looking forward to the future where we're going to be learning about transcription and translation and the transcriptional control. But don't worry about that stuff right now. We're going to focus on replication. All right, so let's dive in. So the big question that underlies all of DNA and that has been asked since before DNA was even discovered to be important was how exactly are the instructions for life, the instructions for building living things, passed from one living thing to another during reproduction, and what is that substance that does that? So that's the big question: is what exactly molecularly is going on that allows for organisms to store information and to pass it on. So until we knew about DNA, we thought that the most likely candidate for a molecule that would be able to have this property of being able to be passed on and also to contain genetic instructions would be proteins. Because proteins were so incredibly diverse, you guys remember we learned about proteins. We said that their function was determined by their shape, and their shape was determined by the order and type of amino acids that were in their polypeptide chains. And those amino acids, R groups, would bind together in different ways based on their chemical properties, whether they were ionic or whether they were hydrophilic or hydrophobic, and that would change the structure of the protein and therefore its function. And since there were just countless combinations that could be made with the 20 different amino acids to make up protein, scientists just thought, yeah, it's a no-brainer. Proteins must be the genetic material. They're the only thing diverse enough to be able to hold all of those instructions, but also they're the only thing with a rigid and repetitious enough structure to actually contain this information. Well, so a lot of scientists were looking at proteins, but something just didn't quite seem right. And so Griffith and Avery in the early 20th century, they decided, well, we're going to do some experiments to see whether or not we can determine what the genetic material is. And so they went ahead and they did some experiments with mice, and they realized that actually it wasn't protein, but DNA that seemed to be what they called the transforming principle, the molecule that was able to pass on traits from one organism to the next. Now, that was a huge discovery, and when the results came out, a lot of people were very skeptical because DNA up to that point wasn't considered to be very important. It was known to be a part of most, if not all, living things, but people were like, well, we don't really know what it's for. So later, Hershey and Chase, like 2 decades after Griffith and Avery, they came along and they were like, okay, we're going to try to definitively show whether or not DNA is the genetic material or protein. And so they did their experiment, which we're going to take a look at in just a moment, and they discovered that yes, in fact, DNA is definitely the hereditary material, not protein. This led Franklin, Wilkins, Watson, and Crick in the 1950s, just after Hershey and Chase, tried to determine the structure of DNA and to see how it would make sense that DNA was the molecular underpinning for all of our genetic traits. So they were able to discover the structure of DNA, and when they built a model of DNA in 1953, or actually maybe it was 54, they were able to show how the structure of DNA does in fact directly correlate with its ability to store information and to be easily passed on. Nierenberg, Philip Leader, and Corona in 1960s learned to read the genetic code within DNA. They learned to translate it into the language of proteins, because if you guys remember, DNA is basically instructions that are eventually translated into protein. And then many scientists in 1977 were able to sequence the first bacterial genome that allowed them to study how a lot of these mechanisms work and gave us the underpinnings for a lot of what we understand today. So this is the history behind the structure of DNA and all the incredible scientists that were involved in it. We're going to talk very briefly about Hershey and Chase because their experiment is the real kicker here. So Hershey and Chase used things called bacteriophages. They're viruses that infect bacteria. And we could see a bacteriophage is a very simple, it's not an organism, it's not living, but it's a very simple molecular machine. It has a protein coat or capsid, which is essentially just a little container made out of proteins. It has these little legs that lock onto the cell, and then it has sort of, you can think of it almost like a hypodermic needle injector to inject DNA or some sort of nucleic acid into the inside of a cell in order to make that cell reproduce more of it. So basically, it squirts its injects, it injects, my God, it injects its instructions into a cell, and then it makes that cell um, read those instructions and carry them out and produce more viruses. So bacteriophages only infect bacteria, and what Hershey and Chase did was really cool. They knew that sulfur was an integral part of the virus's protein coat and their legs and all that outer structure stuff. And they knew that phosphorus was an essential component of DNA because they knew that DNA was partially composed of a lot of phosphorus. And so what they did was they went ahead and they took radioactive isotopes. An isotope is just another version of an element that has a different number of neutrons and subatomic particles. And they took these radioactive isotopes of sulfur and they bound those into these bacteriophages so that their protein coat and their legs and all that outer stuff would glow with radioactivity. It could be detected by a radioactive device, a device that measures radioactivity. And then they went ahead and they did the same thing. They went ahead and marked the DNA or the phosphorus within the DNA with a radioactive isotope so that they could track where the DNA was going. So their big question was when viruses inject whatever their genetic material is into a cell, that genetic material has got to be the stuff that is like genetic material for all living things because that we know that DNA, if it's the underpinnings of all living things, then that should be the stuff that actually goes into the bacteria um, and helps to make more of the virus. If it doesn't go into the bacteria, but protein goes into the bacteria, that would suggest that protein is the stuff that's actually causing the change in bacteria to make more viruses, and thus would be the genetic material. So they wanted to see, does DNA go into the cell or does protein go into the cell? So what they did was they injected two different groups of bacteria. One had the radioactive labeled protein coat, and the other had the radioactive labeled DNA. And they went ahead and allowed these viruses to inject their stuff into the cells, and then they went ahead and they took the infected cells and they spun them down using a centrifuge. It's a machine that whirls these little tubes around really fast. All the infected cells, the bacteria cells, collected at the bottom of the tube, and all the liquid containing the viruses and any other things that were outside of those cells, called the supernatant, was left outside. So the pellets are the bacterial cells that were infected, and the supernatant is all the discarded viruses and whatever else was on the surface of those cells. So they wanted to see whether or not the radioactivity would be inside the pellet or the supernatant, and what kind would it be? Would it be 35S or would it be 32P? What they found was that the 35S, which bound to the protein coat of the viruses, so the outside of the viruses, was found in the supernatant in a large degree. The radioactive DNA was found inside the pellets. And so they concluded from this that because the radioactivity of the DNA was detected inside the cells, that the DNA was the genetic material that was actually passed on to the bacteria. And the protein was just the stuff that was left behind. It's like the discarded little hypodermic needle that's been used and thrown away. So this was one of the big experiments to show that, yes, in fact, DNA was the genetic material. And you can kind of look through their experiment a little bit more if you want to get a better glimpse of it. But it was a really, really ingenious idea. So why was this such a shocking discovery for science of the day? Well, because it suggested, wow, okay, so DNA is actually genetic material. And it also made them ask questions about what about the structure of DNA allowed it to be a better candidate than protein. Nobody knew what DNA structure was. So this led to those four scientists in the 1950s discovering the structure of DNA. Rosalind Franklin was an X-ray crystallographer, which meant that essentially um, she would manipulate crystals to grow around molecules, and she would shine a light through them, and that light would create a shadow pattern that would suggest the general structure of the molecule. Now, of course, it's a very simplified and relatively inaccurate description of radioactive crystallography. If you want to know more, definitely check it out. But essentially what she did was look at a shadow of the molecule to try to see what its shape was. This is, of course, how she had to do it. There was no other way to see something that's small. So her picture revealed four important things about DNA structure, and here's her picture up here, photo 51. The cross at the center of the pattern indicates that the molecule is helical in shape. Excuse me. The angle of the cross showed the pitch or steepness of the helix. The distance between the horizontal bars, so these is right here, showed the number of the turns of the helix, and it showed them to be 3.4 nanometers apart. And the distance between the middle of the pattern and the top of the arms showed that there was a repeating structure within the molecule. Essentially, it just told us, it told them, after some analysis by Watson and Crick, that it was a double helix that consisted of a backbone of sugar and phosphates on the outside and nitrogenous or nitrogen-containing bases on the inside. So here we have that. We have our phosphate sugar backbone, which you guys should remember from Unit 2. And we see on the inside these nitrogenous bases that are all attached together using hydrogen bonds. And so what they showed was that it must be a tightly packed molecule, and that a purine, which would be A or G, the bases A or G, always had to be bound to a pyrimidine, which is bases T and C. And that was because of the spacing that they found between the bands suggested that that had to be the case. It also lined up perfectly so that we had 3 hydrogen bonds between a guanine and a cytosine and 2 between adenine and thymine, which was a very stable structure. And so they determined this is the structure of DNA. It had an anti-parallel or upside down or inverse structure. So one side would go from the 5 carbon end to the 3 carbon end, and the other side would go from the 3 carbon end to the 5. So they were heading essentially in opposite directions. And if you look, what that really means is if we count the carbons, starting with the right hand most, I'm going to use the left hand most arrow in this little cross arrow pattern to show you. So this carbon right here, that's 1, 2, 3, 4, and 5. So we can see the 5 carbon is at the top. The 3 carbon says 5, 4, 3. The 3 carbon is at the bottom. So that's why we say 5 to 3. It ends here with that 3 carbon. Whereas over here, if we count, and we're counting in a similar way, we're going to start here. So we have 1, 2, 3, 4, 5. And so we can see the 5 is on the bottom and the 3 is on the top. So that showed an anti-parallel structure to DNA. It was a double helix with bases in the middle, sugar phosphate backbone, anti-parallel. Now, one thing was really interesting about what they found. When they did that supernatant experiment, they found that there was actually a little bit of protein that was contained within the cells. And so when they looked at the structure of DNA, they were like, yeah, it's tightly packed. It has to be efficient and repeating. That makes sense if it's going to store information. But there seems to be, it seems to be way more tightly packed than even a double helix in most cases. So what later was discovered was that proteins were actually involved. So it wasn't, DNA is just never just by itself. It's actually always with proteins. And that actually makes a lot of sense because, when they're talking about proteins being another likely candidate for genetic material, I mean, proteins and DNA go together, I don't know, like chickens and eggs. I mean, they have to be together. And so that was a really exciting discovery to see that, yes, in fact, proteins do always work together with DNA. And oftentimes how they work together with DNA is they help to tightly wind DNA and wrap it into a very condensed form known as chromosome, which we have learned about in genetics, and you guys kind of know what the underpinnings of that is, that the chromosome is basically like the state of DNA when it needs to be transported or when it needs to not be accessed. And of course, we'll talk about that a little bit more as we go. And so they discovered that, there were these proteins called histones, and DNA would wrap around the histones, and it would wind it up, and it would supercoil it into that very tight chromatin or chromosome structure we saw, so that it wouldn't always be in use. So there are certain sections of DNA that need to be in use. Those are the ones that are unwound, and certain sections that don't be, and those are wound back up tightly, both to protect them and to regulate what genes are made at what times. Okay, so we're going to skip some of the stuff about nucleosomes. We don't need to know that. Okay, so for DNA to replicate without losing its information. So one of the things that they determined about DNA that was really important is that DNA not only had to be able to contain information in its bases, so those repeating units, A, T, C, and G, they were found to be perfect to contain codes to build structures like proteins. So it was already shown that the structure of DNA would be perfect for building stuff. But not only did it have to be a good instruction manual and have a lot of really clear-cut instructions inside of it, it also needed to be able to be replicated. So if you think about it, an instruction manual is useless unless you can either give it to somebody or create copies of it to hand out. If a company had one copy of an instruction manual and yet they had like a million people working for them and needed to do the same task, it would be super inefficient and basically impossible to pass out the same copy to each one. Like, just be like, okay, after you're done with that, Bob, pass it over to Susie, and Susie will pass it over to, that's just not going to work. Everybody needs their own copy. But to do that, an instruction manual has to be easy to copy. And so DNA needed to be easy to copy. It also needed to be durable, because if you think about it, if you're given an instruction manual and it's important to your job, you want to make sure you take care of it. You don't destroy it because you need it. So DNA needed to be durable, it needed to be consistent, it needed to be easily accessible, and it needed to be easily replicated. Those were four really important things that needed to be true about DNA. So today we're going to focus on DNA replication and talk a little bit about what that process actually means. So Meselson and Stahl in the 1950s, you can tell there's a lot of DNA research happening in the 1950s. They came up with this model of DNA replication, how it occurs, called the semi-conservative model. Now that does not mean that it goes out and it votes for moderates or for Republicans. It means that the DNA replicates itself in such a way that is super efficient and requires very few resources. So let's go ahead and take a look at DNA replication and how it manages to do this in a semi-conservative fashion. So this is what DNA replication looks like. I'm going to shift this down because I can see that the click to add title thing is getting in the way. There we go. I'm also going to shift this down and get rid of this because this is also going to get in the way inside. Okay, so here we have DNA replication. I'm going to explain each part of this, so don't worry if you don't understand what's happening. You shouldn't. But we're going to start over here. So DNA, here's our double helix. It has to be unwound in order to be replicated. So remember how I said it was wound into a tight structure called chromatin most of the time? Whenever it needs to be used or replicated, it's unwound. And it's unwound by something called topoisomerase, which is also called DNA gyrase. So this is a protein. Remember I said proteins are always working together with nucleic acids. The protein that unwinds the helix from its supercoiled state and then straightens it out so that it can be used for replication. So the point of DNA replication is to double your information, essentially make an exact copy of your DNA. This is really important if you want to pass on your DNA to another organism, say during reproduction, like we've talked about. When you want to double your DNA during the cell cycle, when you're doing mitosis, or when you're making gametes in meiosis. So that happens, if you remember, during the S phase of the cell cycle. So when we're going to replicate this thing here, what we want to do is we want to unwind it from its supercoiled state, and then we want to open it up. So all of the important parts of DNA are really the bases. That's the information we need to get to, but they're on the inside of the molecule. So to open up the molecule, what we have to do is we have to go ahead and use an enzyme called helicase. Helicase comes in and it's going to break those hydrogen bonds between the nitrogenous bases in the center of the helix in order to open it up. Once it's opened up, these little proteins called single-stranded binding proteins are going to attach onto each one of the single helices. So the reason they do that is they basically stop it from immediately snapping back together. You have to imagine that as helicase goes by, it's breaking apart the hydrogen bonds, but the hydrogen bonds naturally want to reform because they were comfortable that way. So the single-stranded binding proteins stop that from happening. Once that occurs, we end up having something called DNA primase come down. And DNA primase is going to lay a primer. Now, basically, a primer is a landing zone light. So you can imagine it's like at the airports when they have these people like kind of waving around those little lights and being like, land here, land here. It's basically a little landing light for an enzyme known as DNA polymerase. So DNA polymerase, which this is DNA polymerase 3, I don't think you'll need that amount of detail, but I'm going to tell you it's called DNA polymerase 3. It's this big molecule that comes down, and what it does is it attaches onto the single strand of DNA that's open, and it goes ahead and it finds complementary nucleotides. Those are the monomers or the subunits of DNA, and it goes ahead and attaches them onto the open nucleotides to form a new double strand. What do I mean by that? Well, so over here we're still open, right? And so eventually the DNA polymerase is going to come over here and it's going to come up to this base, and let's say that this base is an A. That DNA polymerase is going to come up to this A, it's going to read it as A, and then it's going to grab a T that's floating around. It's going to grab that T, and it's going to come in and lock that T onto that A to T, C to G, remember. And once it locks that T onto that A, it's going to make the other side of that strand, right? So we're going to have a double strand when we're done, just like this guy's going along and he's making a double strand out of this single strand. And so we're basically exactly copying the DNA as we go, which is really cool. So now we should have an identical DNA strand over here and an identical DNA strand over here, which means we have two identical DNA strands, whereas we originally started with one, which is really neat. Now here's where the semi-conservative idea comes in. So semi-conservative just means that instead of DNA looking at the original and making a completely new copy just from scratch. So basically being like, okay, this is the original molecule. I'm going to come down here and just reform a new one. Instead of doing that, it uses the old parts of the original molecule, these original strands, to synthesize the new parts shown here with the green bases, or excuse me, shown here in these double strands, to go ahead and replicate its DNA. So essentially what we're seeing is we're starting off with one and making two based off of half of the old. That's that semi-conservative idea. Now up here, what's happening here with this Okazaki fragment? So Okazaki fragments are formed because DNA polymerase has this really interesting rule. It binds onto the 5 carbon of each one of the nucleotides, and it rolls forward like a train on a track from the 5 prime to the 3 prime end. So what that means is it binds onto the 5 carbon, and then it goes to the 3 carbon, and then gets shuttled off to the 5, to the 3, to the 5, to the 3. And that's the only direction that polymerase can operate in. Now that's fine for what we call the leading strand, which is the strand of DNA that naturally goes from 5 to 3. That's fine for that one. But for the lagging strand that goes from 3 to 5, that's a big problem because DNA polymerase can't go backwards. So what has to happen here is that DNA primase is going to lay down a bunch of primers, and DNA polymerase is going to bind onto those primers and start synthesizing the new strand in the 5 to 3 direction. So basically, this polymerase here is going to attach onto this primer and it's going to go this way. Now, because it's not continuous, right, it's having to attach and go this way, attach and go this way, there are little gaps between these synthesized strands. And we call those little gaps Okazaki fragments because the scientist who discovered them was called Okazaki. And so we have these little Okazaki fragments. And so we have to have this final protein called DNA ligase come in. And DNA ligase is going to go ahead and grab the little gap between those bases and it's going to stitch them together. So basically it's going to put together those little fragments so that we then have two complete strands where we only started off with one. So that's the idea of DNA replication. We've taken one strand of DNA and we've turned it into two new identical strands to the original. So we have two copies of our instruction manual, whereas we started off with one. Now, this is very important, again, for passing on these instructions to newly created cells or passing on these instructions to offspring because they need, a complete set of DNA. So replication it happens all the time in your cells. Now, mistakes can happen, and those mistakes that happen are really easy to fix because your body can use the original DNA strand to check to make sure it was right. So going back to this example here, if this was an A, and my DNA polymerase came along and it accidentally put a G there attached to the A, well, the body's going to come back and scan over it again using a molecule called DNA polymerase 1. And when it comes back and scans it again, it's going to be like, wait, there shouldn't be a G here. It should be a T because it's an A on the original. So it's going to get rid of the G and put a T there, and it's going to fix the problem. There are problems that get through that aren't fixed. It just happens. Mistakes are made. And those problems are oftentimes the basis for mutation. So that's where we get those single nucleotide polymorphisms or those little substitution changes where we end up changing a base in the DNA and it changes our protein, or maybe not. But that's that synonymous and non-synonymous we talked about a long time ago. So this is the process of DNA replication. Here are all the proteins involved and what they do. So I just talked about each one of these. I won't go into these any further, but you guys can look at it through the PowerPoint. And this is the steps that occur in order for this to form. Okay, so again, complications, since DNA polymerase 1 and 3 can only add nucleotides to the 3 carbon of the previous base's sugar, it can only synthesize new strands continuously in the 5 to 3 direction. But because we have something we have anti-parallel strands, one strand will be going against that direction, and so we have to add primers and synthesize in the wrong direction, which creates fragments we have to stitch together using DNA ligase. That's one of the biggest complications. The other one is when we make mistakes. So that's what we mean by leading and lagging strand, just to further clarify. Leading strand is the one that goes naturally from 5 to 3 and can be synthesized continuously. Lagging strand is the one that goes from 3 to 5 and has to be synthesized in little gaps or fragments to be put together later by ligase. So by the end, what we've done is we've taken one DNA strand and we've turned it into two. So that's DNA replication. That's all I'm going to cover today for you guys, but it should give you a general idea of how this occurs. I'm going to add a couple more videos talking about replication to help you out, but hopefully this should inform you as to the structure of DNA and how it replicates itself. Next class, we're going to be learning about transcription. We'll learn about RNA, which is the other type of nucleic acid, and we'll learn about how DNA and RNA are both used in order to make the instructions that will eventually lead to the creation of proteins. I hope you guys have enjoyed this and that this has been informative and useful to you. Enjoy your all's days and week, and I look forward to seeing you again with another lesson on Wednesday. All right, guys, take care.